All right, so last time we talked about making a scatter plot with your PCA data. And let's see here, let's just go back and look at what that plot actually looked like here. Let's do it here. Sorry. Okay, where's my plot? Oh, it's because I'm doing it up there. Here we go. There's my plot. We're going to print it. Boom. So here's our plot over here, and we see some nice clusterings here, but it's kind of interesting that our groups out here are mixed. So one thing you might want to do is go back and see how gene expression is leading to these principal components. So I'm going to show you how to do that a little bit today. So fun fact that you may not have known is, so here's our PCA object that we created from PRComp. So it has a couple different attributes. So you can find those by doing PCA $, and then if you hit tab, it'll bring up some of its attributes. And we've been looking at rotation, but it also has center, it also has scale, and it has this value called X. So if we just look at what that is, I wonder if that'll work with it. Okay. So if we go and look at what that is, what you'll see here is it's got the gene names on the left and the principal components up here at the top, and then it's got a value here. And what this basically tells you is basically how these genes contributed to those principal components difference off of normal. So what we could do to actually figure out how these genes contributed to our PCA plot is we can sort them. So let's go ahead and sort them here. So I'm just going to go do PCA cont for contributing. Oops, copy, I paste. So I'm going to make a data frame and then with that value. And I'm going to sort it based on principal component 1. So now if we look at this, here, let's do head. Save ourselves some pain here. Why don't we do, well, you can see that now we're sorted from least to highest. But you know what we should do is we should order it on absolute value. I think this is how we do absolute values. This will actually be more informative than what I was going to do before. Hold on a second. Okay, so I was trying to figure out how to get it in the correct order. So we want actually the higher numbers first. So now if we look at this, okay, we've got our higher numbers first here in column 1. So we don't really care the direction, we just want to know what's contributing to the spread of that primary component. And so now what we want to do, let me fix that up here so that when you guys get to this, if you want to run it yourself, it'll work out okay. Yes, that's true. Okay. We'll just copy and paste the one I know worked up to there, decreasing, not descending. Okay, so we'll run that. And now what we're going to do is we're just going to grab the top 100 genes that contributed to this. So we're going to say top 100 contributors equals head, the row names of PCA contributors, comma 100. So we just want the names. So we've got those there. Now we've got the counts data already loaded from before. So what we're going to do is we're going to just grab the count data from those genes that contributed the most. So contributing counts equals counts. And then we're going to do row names of counts that are in that top 100. And so now we do this. Got an extra scene there. If we do this, now you see we've got the count data for each one of our samples here and the associated genes. So if we ask, just double check ourselves, we'll say length row names of contributing counts. Say we've got 100 of them. Okay. So one thing to remember is we've got these kind of jacked up names here. So if you remember how I did in the last video, it was kind of confusing. I used sapply and everything. Realized I was doing that way harder than I needed to. So we're just going to say call names of contributing counts equals G sub, get rid of the MCF7 because everything's MCF7. Replace it with nothing for call names of contributing counts. And now if we go look at those new call names, boom, that's gone. And now we're also going to get rid of the demute and replace it with the correct mutant names. And the ymute, correct the mutant name there. This one I'm not using at the moment. All right, so now it's not perfect, but it the names are at least somewhat consistent. So now what we can do is we can use pHeatmap, which is a popular heat mapping program. And what we could do is we could just drop our data frame in there. And you see this heat map on the right. It's not terribly informative. Pretty much everything is got a low value except for a few in the corner and the top corner and the bottom right corner. So what we're going to want to do is we're going to want to add scaling by the rows. So basically, whether it's high or low, it only is looking at each gene individually, because you may have a really highly expressed gene and a really lowly expressed gene. So all the, say, the highly expressed genes are going to blow out everything here. So we just want to say, looking at each gene for each sample, how high are they? So we'll say rows, scale equals row. And now you can see, okay, this is looking a little more interesting. Now we can sort of see clusters. We're going to do a few other things where we'll say pHeatmap. If you're not sure what all your options are, if you go into it and you hit tab, it brings up every possible value that you can set. So you can see there's quite a few here. The matrix is the first thing we put in, so that's our contributing counts. Scale is equal to row. And then I'm kind of annoyed by these the borders. So we can set border color equals NA. Now, how did I know that? That's a good question. So real quick, if you are inside the function, you can hit F1 and it'll bring up the help for it. And I can just say border color. And what you see here at the top of this usage is the default value. So if you don't put a specific value in for it, this is what it'll automatically do. So this is the default color and everything. So it says the border color equals gray 60. But here, if you go down to the arguments, it has help for what you can do with them. It says use NA to make them not drawn at all. And I think that's what we want to do. So here we go. Here's our new thing. And now you can see we've got a cluster down here. And we've got a cluster up here and maybe a third one here. So you've got the trees, the dendrograms here. You can turn those on and off. You can change a lot of values. One thing that I'm seeing over here that's not particularly useful is all of the font names together. So I'm just going to say cell height equals 10. And now you can see, hey, I can read every single name here. But the problem is now our thing is too large. And after screwing around with this for a while and even asking on Stack Overflow, I think the easiest way to figure out what the right size for this graph is just to do PNG. So this is making a PNG image. So let's do this real quick. So we say PNG, we give it a file name, so pheatmap.png, and then we say height. The default height is 400 pixels, so there you see it there, the 480 pixels high and 480 wide. Obviously, we need to go a little bit higher, probably quite a bit higher. So let's just say what happens if we make it 1,000 pixels high, and it's going to want me to give it a width. So we'll keep the default width because that seemed pretty good. And then once we save the image, so we've created an image container, now we have to put something in it. So we run our pheatmap and then we say dev.auth, and that saves it. And so this is the file. Let me see here, let's make sure I've got the most recent one here. Okay, so there's the graph it made. Graph is still not tall enough. So what I'd like to do is I just leave it open and then I just go back and modify the values. So the height, let's go to 1600 pixels. We run it, we turn it, save it. Okay, now we can see we overshot it a little bit. I think it's like 1300 might be good. Okay, so that's about right there. So there you go. That's how you make a nice heat map. You can do other things like turn off the row names if you want, but I think in this case we're actually interested in what genes are contributing to what groups. So you can see you've got the long-term estrogen deprivation group here on the left side. You see they're all clustered together independent of whether they are a wild type or one of the mutants. So you can see, okay, these top genes are probably contributing. Maybe you could even say down to here that these genes are up in that group and these genes are down. I'll tell you about this real quick. So you can see as we zoom in, some of these, the text is kind of hard to read. If you're making this for, say, a web page, if you're not making it for a web page, you should save it as a PDF. So if we look at, let's see here, PDFs don't want their heights in pixels. They want their height in inches. And I think I figured out that this was about 14 by 4. Oh shoot, I saved the name as a PNG even though it's a PDF, so let's fix that real quick. PDF. So now if we go and we look at it, I didn't get the height quite right still. So this is one annoying thing. Normally it resizes it for you automatically, but since I'm setting the row height, it won't do that. So you're going to get to see me fiddle around with this a little bit. Okay, there we go. So here, if you make a PDF, the fonts all look much nicer. You just have to get the widths right. if you're making it for a web page, you can't really show PDFs very well on a web page. They're kind of annoying. You have to do an iframe, which to most of you that won't mean anything. So if you want to make something like a PNG, so a PDF is a vector, which means the resolution's the same no matter how much you zoom in, because it's basically a mathematical formula telling it how to draw this thing. Whereas a PNG is actually a pixel value. So if it's low information content, then it'll look pixelated. So what you can do here is the res is the resolution, so it's dots per inch. So that's normally set to 72. Publication quality is around 200 to 400. So if we set it to 400 and then say units equals inches, that's for the height and the width. So let's go ahead and make this. So we know those heights and widths should work okay because we already did it with the PDF here. If we go over and peek at it, you got everything. All right. Now if we go look at this, when you zoom in, everything's a lot crisper. Makes a bigger file, but that's okay. So clearly we need to make it just a hair wider. Let's just make it 5 inches wide. And now if we go look at it this last time, we've got ourselves a fairly nice graph that you could feel comfortable putting on a website for somebody to see or sending to somebody. So there's a ton of stuff you can do with pHeatmap. I'm not going to go into it all, but like I said, if you just bring up the pHeatmap function and you hit F1 while you're over the top of it, it'll bring up all the different possible values that you can set, such as the number of tree heights, the number of like splits you get between the trees. So like if we wanted it to cluster slightly different, so let's see here. I think this is one thing that's not a terrible idea. So you use the cut tree, so the number of clusters that it's divided into. So this is kind of interesting. So if we go and look at our graph here, let's say for the columns, it's got 1, 2, it splits into trees for every single group, but we only say want it to split into the treatments. So we've got estrogen, DMSO, and long-term estrogen deprivation. So we'll try and do cut tree calls equals, we'll say 3. Why don't we save this as a file so we can actually look a little bit closer at it. Let's try this again. Okay, so here's our graph here. So here it's split it into 3 groups, and it puts a little line between them. So now you can see that we definitely our long-term estrogen deprivation, it splits into 1 group, whereas most of our wild types go into the 2nd group, but we've got a single wild type outlier. So it'd be interesting to look into this guy and be like, why are you so screwy? But if you look at the splits here, you can see the thing that's probably making these wild types split into their own group is this cluster here. And for some reason, even though this guy has some pretty high values in that cluster, for some reason it just wasn't above the threshold. So there's probably some other things contributing to it. So that's just some things you can do. Obviously you can mess with the graphics, you can change the colors, you can add labels for your axes, you can just get rid of the clustering, you can just get rid of the graphics for the clusters, you can change font sizes, etc., etc. But I hope I unpause this, and I hope that this was informative.